series of forensic science lecture. Today we are going to discuss about human genetics. We are studying human genetics. It is an essential part of forensic biology and forensic genetics. The application of human genetics is very well used when we find any biological material on the crime scene. From that biological material, we extract the DNA and we can go for the genetic study, which will be helpful in many ways. To understand the forensic genetics, we have to study the basics of human genetics. To go about what is human genetics? The major definition is, it is the study of how human traits are passed down through generation. The traits when we are talking about are those traits which makes a human being. For example, the height of the person, the eye color of the person, the skin color of the person, the hair texture of the person. Major traits may be which are visible to the human eye and which are not visible to the human eye. They are passed down through generations. It is a branch of biology that includes many overlapping fields. For example, we can name population genetics, medical genetics, clinical genetics, epigenetics, genomics, etc. So, whenever we are using any genome studies, we can consider them as genomics. If there are any epigenetic studies, then we can call them as forensic epigenetic study. Any study which is being related to health or something, they can be categorized under clinical genetics or medical genetics. So, importantly, categorically, if we see any application of all these fields, maybe they are molecular genetics, population genetics, medical genetics, epigenetics, genomics, when they have application in the field of forensic or in the field of any crime, law, then together we call them as human genetics or forensic genetics, which has further applications. So, going towards human genetics, there are certain terms which are being used in this field in day-to-day life. We should know what are these terms. The first important term which is considered important, which is in the field of human genetics, is genetics, which is the study of inherited traits. So, genetics is the study of inherited traits. This genetics can be studied in the context of humans, animals, plants, etc. If we are studying about the genetics of any plants, we can call it as plant genetic studies. If we are studying on animal model, then we can call as animal genetic study, and specifically if we are studying the inherited traits of humans, we call them as human genetics. So, in forensic science, the major purpose where these genetic studies are being used is the identification of an individual, where we look for many polymorphisms, or in the case of mass disaster cases, or maybe in the cases of the paternity cases, where we have to identify whether the child belongs to the father or not. So, these inherited traits are studied in many cases where we are considering the forensic genetics. So, when we talk about genetics, the major term is chromosome. Chromosome are rod-shaped structure which are present in the nucleus of the cell, and that contains the material DNA. DNA is deoxyribonucleic acid. This is the major genetic material which serves to convey inherited traits from one generation to another. So, how DNA is being transferred from one generation to another? By mitosis, meiosis, it is being transferred through generation to another. Then the next term is locus. It is a place on a chromosome which is occupied by a single gene. So, we talk about various genes. These genes are present on DNA, and a specific place on the chromosome we call as locus. So, why do we need to study these locus? For example, if we are studying the forensic genetics and we have to identify an individual and we are going for polymorphic markers. The polymorphic markers, we are talking about STR markers, VNTR markers. These STR, VNTR markers, we have to define them by a name. So, they are defined that this particular STR marker is located at the particular place on this gene, on this chromosome. So, that is why we need to study the locus. that at this particular locus on the chromosome number, for example say 10, on this gene this particular STR marker is present, which is further help us in identifying any individual. So then we have two terms, gene and allele. Gene is the unit of heredity material, whereas allele, it is the alternative form of a gene at the same locus. So let's have a look at this picture. It defines where we can see that two chromosomes are there, and at the particular location of those two chromosomes a gene is present. There are 30,000 genes around on our DNA. So where we are talking about any one gene. So any one gene, it can have two allelic forms. For example, let's talk about the height. If it is the gene which represents the height of a person, then it may have two allelic forms, which is representing as a tall height and a smaller height. For example, allele 1 says capital T, which represent that this is dominant, and if the person will have a capital T, person will have a taller height. On the other hand, if we talk about the allele 2 says that it is small t. Small t means the smaller height. So if a person will have small t, then he will have a smaller height. So gene is coding for a specific trait. That trait could be anything. Alleles are the two forms of that gene. So which says that a person can have more than one form. A person can have a taller height or a person can have a smaller height. This is just an example to understand how gene and allele are related to each other. So when we talk about the gene and allele, the allele I said, for example, we are saying the example of the height of the person. So capital T says that it is the height for tall, and small t says it represents the height for a smaller person. Now comes the two terms, homozygotes and heterozygotes. Allele never exists singly, the allele exist always in pairs. Why? Because we are diploid organisms. Diploid organism means that we inherit one chromosome from our mother and one chromosome from our father. So if we inherit one chromosome from our mother, so one allele will come from the mother and one allele will come from the father, depending upon what allele is present with the mother and what allele is present with the father. If similar alleles come from the mother and the father, for example, the father is also giving allele of capital T and the mother is also giving the allele of capital T, that means the person will be a homozygote having the two same alleles, capital T, capital T. So if the father is giving the allele of small t, mother is giving the allele of small t, then the person again will be homozygote with small t and small t. So the homozygote could be, it has to be similar. That means having the identical allele at the corresponding loci on a pair of homologous chromosome. So if a person has capital T on both the chromosome, we call that person as a homozygote. Whereas if the person has different alleles, for example, the person inherited capital T from the mother and small t from the father, and the person is having two different alleles, capital T and a small t. That means having two different alleles at the corresponding loci on a pair of homologous chromosomes, we call the person as a heterozygote. Homozygote will have the similar alleles and heterozygote will have two different alleles. So talking about this, depending upon which allele is inherited by the person, that determines the genotype of the person. We say the capital T allele is the dominant one. Dominant means that that allele will express itself. So if a person having capital T or having a capital T and small t, the person phenotype will be the taller height. So genotype might be different, but it is giving a same phenotype. But a person is having two recessive alleles, that means small t, the person will have a smaller height. So, genotype tells us the actual genetic composition, whereas phenotype is telling us about the observed expression of the genetic composition. So, taking that example back, genotype telling that the capital T, a taller height, capital T and small t combination are heterozygote, the phenotype will be same, the taller height. So, phenotype is what we are looking from the outside. We say the phenotypes which we use in forensic investigations, different phenotypes we look for. For example, if a crime is being committed and a person identifies the accused, he says he has a complexion of around fair, he has a height about this, the hair color was grey or black, the eye color was this, the nose shape looked like that. So, giving the description how a person looks like from outside determines that is the phenotype of the person. Genotype could be anything which is telling about the phenotype. So, a phenotype comes from the dominant allele, the allele which is being expressed inside. So, the allele which always express itself in the phenotype, whether it is homozygous condition or the heterozygous condition, we call it as the dominant allele. Now, when we are talking about dominant allele, another term which we talk about is recessive allele. So, looking at the picture, genotype versus phenotype. The genotype is an organism's genetic information, as I have given the example of the height. Looking this example, capital B is a homozygous dominant, capital B small b is a heterozygous, but small b is homozygous recessive. So, that means whenever capital B will occur, that time the condition will be dominant and the flower will have a purple color. So, you can see the corresponding picture that the phenotype is the set of observable physical traits. If there was capital B homozygous dominant, then the flower color was purple. Even in the heterozygous condition, genotype is different, but the phenotype is purple only because capital B is giving purple color to the flower. In the absence of capital B, when we have small b, that means homozygous recessive condition, the color of the flower becomes white because there is no dominant allele present to give purple color to the flower. This is how genetics of genotype versus phenotype works. We have the homozygous dominant, we have the homozygous recessive. Depending which of the allele is expressing itself, that will give color to the flower, or in humans that will give the phenotype to the person. As we were talking about the various genes, they give the skin color. So, if we have a fair color or if we have a dark complexion, that is our genotype. Depending upon which genotype is being dominantly present in our body, that will give different facial features or different phenotypes to the humans, which we study in forensic science. Now, going to the dominant allele and to the recessive allele, when we were talking about the dominant allele which is expressed in any condition, even if recessive allele is present, but recessive allele is expressed only in the homozygous condition, as we have seen in the previous picture, that the white color of the flower only came when it was a recessive homozygous condition, small v and small b. When even a trace of capital B was present, the flower got the purple color. So, if we are talking about the dominant and recessive, there also another condition, codominant. That means alleles at a locus express themselves in the presence of each other. So, looking at the example, the dominant color is the red of a flower, recessive is the white of the flower. But when a condition occurs, codominant condition occurs, in such case the red color and the white color, they both have equal powers. So, the flower becomes a combination of red and white. We call this condition as codominance, where the red and white color is being both the genotype is present of both the dominant and recessive, and the phenotype of the flower came out as the red plus white. But when there is incomplete dominance, then it becomes a mixture of red and white and it will give us the pink flowers. So, here we understand the four concepts: dominant, recessive, codominant and incomplete dominance. Dominant, it will be red only; recessive, it will be white; codominant, both they have red plus white; and incomplete dominance, that it becomes a combination of red and white. So, when we are talking about different traits, different genes, these genes are present on different chromosomes. So, we have in humans, we are studying the human genetics, we have 23 pairs of chromosomes. Why 23 pairs? Because 23 chromosomes comes from our mother and 23 chromosomes come from our father. So, we have in total 46 chromosomes, but we call as 23 pairs. Out of these 23 pairs, 22 are autosomal and 23rd chromosome is the gender revealing or the sex chromosome. It determines that what will be the gender of the child. So, we call the traits which are present on the autosomal chromosomes, they are known as autosomal traits. Trait carried on any chromosome except a sex chromosome are autosomal traits, but the traits which are present on the sex chromosomes. So, what are the sex chromosomes? The sex chromosomes are XX in female and X and Y in a male. So, depending upon whether the Y is being transferred, then the child will have the male gender. So, if the father transfer the X, then the child will have the female gender. So, those traits, the sex chromosome defines the gender of the baby. Autosomal traits, they are present on the 22 pairs, but the sex chromosome is the 23rd pair. When we talk about all these things, there is certain terms, the gene pool and the Mendelian population. Gene pool determines the total of genes which occur in a breeding population. So, population we say as the Mendelian population. Mendelian population we call it because the term came from the Mendelian genetics. The scientist Mendel, he has certain law of genetics. He defines the law of hereditary, how the genes are being transferred from one generation to another. We can see, actually we call it that there are certain traits present in the father and the mother which is being transferred to the child, and the child further when he marries a partner, then the traits are being transferred to the next generation. So, this is the Mendelian population. That means they are about a particular caste, about a particular community, and they reproduce inside the community. So, the reproductive community of individual who shares a common gene pool. Gene pool is the total of genes in a breeding population. So, whenever we study any Mendelian population, so we study always go for a specific community or always go for a specific caste, because the gene pool they share is common. The Mendelian population, they follows the laws of Hardy-Weinberg equilibrium, that the particular allele frequency will remain 1 if we look for various alleles in that particular population. This Mendelian population and the law of Hardy-Weinberg equilibrium is also defined by natural selection. Now, what is natural selection? It is a process by which the environment eliminates less adapted members of a population by differential reproduction. That means what gene, what allele or what trait is advantageous to that particular population will remain and will be carried forward to the next generation. But those are not advantageous to the population, those will be left behind. Only those alleles which have some advantages, they will be carried forward to the next generation. This is the law of natural selection. So, when we talk about the gene pool, we say the total of gene in a particular population, in a particular community breeding population. And the Mendelian genetics, these traits, they are inherited from the combination of the chromosome of the mother and the father. So, mother and the father, there are certain traits which are being transferred to their children. Depending upon which trait is being transferred to the children, it will tell the genotype of the person and that will further tell the phenotype of the person. For example, let us see the parents, they have heterozygous phenotypes. When they have heterozygous phenotypes, there is a possibility that the children will carry or will take the traits of their parents having different alleles. That is why in certain cases, being a layman, we say that these are two brothers but they do not look alike. What is the reason that the two brothers which are real brothers, they are from the same parents but they do not match, their phenotypic features do not match? The simplest reason is because their genotype is different. They have taken the traits from their parents where one of the child has taken different traits and another child has taken different traits. That is why the parents, if the same siblings are there having the same set of parents, they will not match because of the different genotype and there will be a different phenotype. It will be defined on the basis whether the dominant allele is being transferred to them or the recessive allele is being transferred to them. So, when the parents, they have heterozygote traits amongst them, their child may be looking differently. So, depending upon, if we talk about various diseases in terms of clinical genetics or medical genetics, there are certain times when the disease is being transferred to one of the kin and not transferred to another child. So, that is along the same lines that certain traits which are inherited according to Mendelian genetics might be transferred to certain children and might not to another children. We the humans, being diploid organisms, we have paired homologous chromosomes. That means we contain two copies of each gene, which is we are calling as allele. As we have discussed in the previous slides that supposedly we are talking about the gene of the height. We receive the capital T from the parent and the small t from the mother, capital T from the father. So, a combination of capital T and small t which I received from my parents, being the capital T dominant, I have a taller height. But if I have received small t from my father and small t from my mother, then I have a smaller height because I am a homozygote recessive. So, depending upon what trait or the allele I am receiving from my father and what I am receiving from my mother, it will tell us how my phenotype will look like. This we call as the genetic polymorphism, that the occurrence together in the same locality of two or more discontinuous form of a trait. For example, there can be different kind of polymorphisms. When we are talking about the genetic polymorphisms, we look for the single nucleotide polymorphisms, we look for the STRs, VNTRs, where we have the whole DNA sequence, we look how they are occurring polymorphic in nature. These genetic polymorphisms are a major way to detect the identity of a person in the case of forensic science. So, to conclude this, I would say genetic polymorphisms, which is the occurrence of the alleles in the same locality of two or more discontinuous form of a trait helps us in the identification of the individual, and in the today forensic labs we are using this genetic polymorphisms in the form of STRs, which are the gold standards, and they help us in the identification of the individual. Thank you.